The overall goal of the following experiment is to detect IgE-mediated drug hypersensitivity by the Basophil Activation Test using well-characterized, haptenized drug carrier protein conjugates. This is achieved by first chemically linking a drug via an intrinsic or organochemically introduced carboxy group to lysine residues of human serum albumin. Secondly, the conjugates are physicochemically characterized to determine the exact coupling degree by performing high-performance size exclusion chromatography, or HPSEC, sequential refractive index, and ultraviolet detection. Next, the well-characterized drug conjugates are used to stimulate basophils of drug hypersensitive donors. Ultimately, the Basophil Activation Test, or BAT, can be used to evaluate IgE-mediated mechanisms in drug hypersensitivity.
My name is Markus Steiner from the Allergy Research Group at the University of Salzburg in Austria. Today, we would like to introduce a technique to you which has the implication of measuring the involvement of drug-specific IgE antibodies in drug hypersensitivity reactions. Here we show the procedure with two widely used representatives of non-steroidal anti-inflammatory drugs, namely propionate and diclofenac. However, the method has the potential to be used for routine diagnosis of immediate type drug allergies in general.
Add 10 microliters of a freshly prepared N-ethyl N'3-dimethylaminopropyl carbodiamide hydrochloride, or EDC stock solution, to the propionate or PP sample. Vortex the sample for 1 minute. Next, add 16.9 microliters of human serum albumin, or HSA solution, with a concentration of 118 mg per milliliter to the sample, and then incubate the sample for 2 hours at room temperature while shaking at 300 rounds per minute. After shaking is completed, dialyze the samples 3 times against 2 liters of PBS for several hours each at 4 degrees Celsius. Then, centrifuge the samples for 10 minutes at room temperature at 14,000 times G, and collect the supernatant containing the NSAIDs conjugated to HSA. Check the supernatant for proteins by performing an SDS page using 12% gels, loading about 12 micrograms of HSA conjugate into 1 gel slot. For the analysis of the coupling rate of the NSAID conjugates by high-performance size exclusion chromatography, or HPSEC, first dilute the HSA standard sample to a concentration of 1 mg per milliliter in demineralized water. For detector calibration, inject 50 microliters of HSA standard solution. Then, using a 7.8 by 300 mm TSK gel G2000 SWXL column and an online coupled refractive index, or RI, and UV detector system, calculate the RI constant K sub RI and the UV constant K sub UV for the detector using these two formulas. Dilute PP derivative standards for HPSEC in demineralized water in 5, 10, and 30 nanomole amounts for ease of injection. Then calculate mean values for dN by dC of the drug and dA by dC of the drug for both standards using these two formulas. Now, inject the drug conjugates onto the HPSEC and determine their RI and UV peak areas. Then, using the same two equations, calculate the concentration of the PP derivatives and the HSA for the unknowns. First, label 7 flow cytometry vials depending on their content: unstained control, negative control, positive control, and drug conjugates. Next, pipette 50 microliters of basophil stimulation buffer into the vials reserved for the unstained and negative control. Add 50 microliters of the anti-Fc epsilon R1 solution from the Flow2 cast kit into the positive control vial. Now, add 0.02 to 20 micrograms per milliliter of the PP conjugate to the drug conjugate vials in a 1 to 10 dilution series, and then add 100 microliters of stimulation buffer and 50 microliters of the patient's EDTA whole blood to each tube. Mix the samples carefully. Then, pipette 20 microliters of the Flow2 cast staining reagent containing PE-labeled anti-CCR3 antibodies and FITC-labeled anti-CD63 antibodies to each of the drug conjugate and positive and negative control tubes, and mix again. Incubate the samples for 45 minutes in a 37°C water bath. Then, to stop the reaction, place the samples on ice for 5 minutes. Next, lyse the erythrocytes by adding 2 milliliters Flow2 cast lysing reagent to each vial. Incubate the samples in the dark for 10 minutes at room temperature. Then, after pelleting the samples for 5 minutes at 500 times G at room temperature, carefully decant the supernatant. Resuspend the cells in 500 microliters Flow2 cast wash buffer and store the samples on ice until flow cytometry. Use the unstained sample to adjust the flow cytometer's voltage settings. Then, gate the cells predicted as lymphocytes within the side scatter, forward scatter plot, similar to this sample figure. Basophils are first identified as CCR3 high, side scatter low, similar to this scatter plot. After gating the cells into the FITC-PE plot, activated basophils can be further identified by their expression of FITC-labeled CD63 positivity. After setting the cutoff to a maximum of 2.5% of basophils as CD63 high in the negative control, a sample can be considered positive for basophil activation if greater than 5% basophils are CD63 high and the MFI of the sample divided by the MFI of the negative control exceeds 2. This method evaluates BAT as a beneficial tool to detect IgE-dependent drug hypersensitivity. Protein conjugates of NSAIDs are used for activation of basophils. By HPSEC with RI and UV detection, aggregation state, coupling degree, and effective yield of conjugates are determined. As depicted in this figure, 43.5% of the DF conjugates remained monomeric with a coupling degree of 5.0 DF to HSA. For the aggregates, a coupling degree of 9.5 was determined. As depicted here, the PP conjugate was shown to consist of 68.5% monomers with a coupling degree of 21.2. The coupling degree of the aggregates was 27.9. In total, the determined coupling degree of DF conjugates was 7.6 DF, and that of PP conjugates was 23.2. BAT, performed with conjugates under optimized conditions, allows investigations of IgE-dependent reactions in NSAID hypersensitivity. As shown here, only the PP conjugate was able to trigger basophil activation, visualized by an upregulation in CD63 surface expression. 20.6% of basophils were activated as characterized by a log shift in fluorescence intensity, and the MFI increased by a factor of 4.9, from 386 for the negative control to 1893. In contrast, using the DF conjugate, only 3.1% of the basophils were activated, which was below the 5% cutoff. Also, the MFI increased only by a factor of 1.1, from 197 for the negative control to 210. The assay validity is evidenced by 1. Less than 5% basophil activation in the negative control, 2. A log shift in fluorescence intensity of a basophil subpopulation, and 3. Greater than 50% activated basophils for the positive control.
After watching this video, you should have a good understanding of how to perform basophil activation tests and, most importantly, how to differentiate IgE from non-IgE mediated drug hypersensitivity reactions. Furthermore, you should have an understanding of how to expand the applicability of the Basophil Activation Test for drug allergies beyond its already established use in pollen, food and insect venom allergies.
This study discusses BAT is a well-established, although not yet routinely used method for diagnosis of IgE-mediated allergic disease. For drug hypersensitivity, however, its applicability is compromised as low molecular weight compounds are not able to cross-link IgE receptors, a prerequisite for basophil activation. Therefore, drugs under investigation need to be covalently coupled to suitable carrier proteins, e.g. HSA. Important factors for mastering the described technique. Quality of blood sample, less than 12 hours since blood collection, number of detected basophils greater than 500, and optimization of stimulation conditions, time, dose. In addition, so-called non-responders who do not even react with the positive control, antibody directed against the IgE receptor, have to be identified. Therefore, validation criteria have to include an anti-FCRE antibody as positive control. Thank you for watching. Subscribe if it is useful.